So that means the person loses their ability to talk and communicate. And hand use, so that means the person start making repetitive hand washing motions or tapping motions. Slow progress of the brain and head growth. So that means the person, their body still grows normally, whereas their head stops growing at a stage. Gait abnormalities, which means the person loses their ability of walking or moving along on foot. Seizures and mental retardation. Objective. Bioinformatics came about from the Human Genome Project, and the Human Genome Project is an international research that intended to identify all the genes, all the human genes there are, and record the positions on chromosomes. And Rett syndrome relates to bioinformatics because Rett syndrome is an inherited disorder where the mutation is found on the X chromosome, which is part of the human genome. And bioinformatics is also a way to organize, search, analyze, and store all the millions of DNA gene sequences on the computer. And
can you hold the pointer?
Yeah.
And bring your mic closer.
Okay, and this is a picture of the X chromosome, and it shows where the mutation is located, which is at the X-Q2A site of the chromosome. Okay, history. In 1966, Rett syndrome was identified by an Austrian physician by the name of Dr. Andreas Rett. And in 1954, Dr. Rett first noticed two girls as they sat in his waiting room and observed these children making the same repetitive hand washing moves. He then compared their clinical studies and discovered that they were very similar to 6 other girls. Unfortunately, he was unable to discover the gene that causes the mutation because he passed away in 1996. However, researchers continued his research, and to acknowledge and honor Dr. Rett, they named the disease after him.
Alright, Rett syndrome is caused by the MECP2 gene, and the gene is located on the long arm of the X chromosome on the 28th site. And you only need one of the X chromosomes to inherit the disorder. And that's a picture of the mutated gene, and the spots are the different sites of amino acids that can be mutated. Rett syndrome is an X-dominant disorder, and it's exclusively found in girls. And so, since females have 2 X chromosomes, if they inherited one chromosome, then they have enough protein on that other chromosome to survive. And 70 to 80% of girls with the diagnosis for Rett disorder have the MECP2 genetic mutation. MECP2 is a member of a family of proteins called NBD, and the other members of the family are NBD1, NBD2, NBD3, and NBD4. The MECP2 gene provides instructions for making the MECP2 protein, and it's critical for normal brain development. The MECP2 protein plays a role in forming connections between nerve cells, including regulating other genes in the brain by switching them off when they're inactivated. A missense mutation occurs in the MECP2 gene, and only one of the amino acids need to be changed in order to get the disease. And that's the structure of the protein, and again, those are the sites that can be mutated.
Okay, this picture demonstrates the binding of the MBD domain, and the green figure is the DNA, while the blue figure is the protein, the MBD1 protein, which is one of the family members of the domain mentioned earlier. And the two circles on the two purple circles on the protein, that it basically demonstrates how when the protein is binding with the DNA, exactly at those positions, that's where the DNA will make contact with amino acids. Okay. This is where our experiment. This is where we incorporated bioinformatics in our research. Our goal was to understand the alignment of the MECP2 sequence from different species using NCBI BLAST, or in other words, the National Center for Biotechnology Information Basic Local Alignment Search Tool Program, to look for where the mutation is in Rett syndrome will show. Procedure. We did this by first we had to obtain the RNA sequence of the MECP2 gene using the NCBI website, and we needed the RNA sequence because the mutation occurs in the protein, and proteins contain amino acids, and RNA translates nucleotides into amino acids. And then we blasted, after we got our sequence, we blasted the sequence and to receive the different sequences that the MECP2 will compare. And BLAST is a database that's designed to explore all of the available genes there are in their database. After the results from BLAST, we analyzed them, and finally we further narrowed down our search by comparing the human MECP2 sequence with 3 other organism sequences, which were
And slow it down.
After we received the sequences from BLAST, we analyzed them, and we further narrowed down our search by comparing the human MECP2 sequence with 3 other organisms, which are the mouse, the chicken, and the X. flavus, which is the African cloud frog. Okay, and then this was our result. Okay, the red lines that ranges from 0 to 100, it basically just demonstrates that the sequence was exactly the same with the human sequence, the human MECP2 sequence that was used to compare.